Good morning or good afternoon, everyone. And it's my great pleasure to host my colleague Yonghao here for today's SocialDNAing. And just before I introduce Yonghao, so I want to thank my colleagues, John and many others in Colombia, as well as Cesar and Thomas, to keep the SocialDNA floating in the past 4 years. And next week, we'll have Burin Shrang from University of California at San Francisco, and he's going to talk about DNA repair in the neurons and its relation with aging. So Yonghao graduated from Fudan University, China, with a major in chemistry, and then pursued a PhD degree in UC Berkeley with a focus on chemistry and also its applications. It is when he started his postdoc with the Gigi's lab in Harvard Medical School, where he shifted to post-translational modification, started with phosphorylation and mTOR pathway. And then when he had his own lab in UT Southwestern, he developed this chemical approach to really characterizing PARylation site on proteins, and which I think he's going to tell us more today. And then we were fortunate to recruit him on 2022. 22? Yeah, 22 to Colombia. So now he's talking just across the street from me. So thank you, and go ahead.
Okay, so let me see if I can share my screen here. Everything good?
Yes.
Okay, awesome. Yeah, thanks, Shan, for the kind introduction, and also thanks, Shan, and also Eric, for I guess the invitation and the opportunity to speak here. It's a really great pleasure. So I think as Shan mentioned, we the central focus of our lab is really kind of focused on understanding the regulatory mechanisms of protein post-translation modifications. And in the last about 10 years-ish, we've been developing this very strong interest in understanding PARylation, as we know is a central regulator of DNA damage response. I think I presented some of our previous work at the SocialDNAing. That was probably like a couple of years ago, but we recently actually had a couple more recent stories, which I would like to share with everybody. So today I will be talking about those stories. So this is my disclosure. So again, just a little bit background. So I think we don't have to talk about too much kind of background in this audience. We know that PARP1 and PARylation actually really plays a very important role in regulating DNA damage response. As we know, the PARP1 is a DNA damage sensor. It gets recruited to the DNA damage site, and then once it becomes activated, utilize NAD as a cofactor to catalyze this amazing post-translation modification PARylation. Now, obviously, PARylation is functionally very important. It does many, many things, but one of the roles for PARylation is to help recruit all these DNA repair factors and complete the entire cycle here. There is another very important concept, which is PARP trapping, which I will be talking about actually quite a bit in later slides. So this pathway obviously is very important from a molecular biology pathway perspective. At the same time, it actually is involved in many disease contexts. As we know that it's actually critically involved in cancer. But at the same time, when this pathway goes awry, it's also involving ischemic peripheral injury, neurodegeneration, and some of the other metabolic diseases. And this actually has been really kind of built up this very important foundation that PARP1 is really a multifaceted enzyme. Does many things, catalyze PARylation, but at the same time, it is also a major regulator of metabolism. As we know that PARP1 is such an abundant enzyme, it's a very robust enzyme. So when it's activated, it causes a quick NAD depletion and energy crisis and all the other cell stress responses. PARylation actually is a stress signal molecule as well. It's involving biomolecular condensation. It regulates cell death. It regulates immune responses. So this pathway obviously is fundamentally important, but also very critical in many different kind of disease contexts. So obviously, the rationale for developing PARP inhibitors for cancer has been really kind of propelled by this, observation that, there is synthetic lethality between BRCA mutations and PARP inhibition. Indeed, there are actually 4 PARP inhibitors that have been approved by FDA in the last 10 years-ish. I mean, I think that was really kind of a fundamental change in, how we care for, breast cancer patients, ovarian cancer patients, pancreatic cancer patients, and prostate cancer patients with BRCA mutations. And obviously, PARP inhibitors are actually actively being studied in other cancers with homologous recombination deficiency. So, we have been actually extremely interested in understanding the mechanism of action for PARP inhibitors, because we realize that, even though these compounds actually work extremely well in the clinic, the mechanism of action, or the fundamental mechanism of action, is still kind of, incomplete understood. So I'm going to really talk about 2 large kind of paradigms or programs we have been working on in the last 10 years-ish, trying to understand, one is the PARP1 inhibition mechanism, and the other is actually the PARP trapping mechanism. And again, just coming back to the enzymology for PARP1, as we know that, PARylation is catalyzed by PARP enzymes utilizing NAD as a cofactor, and the ADP ribose being conjugated to the proteins. And we know that this is work we have been, published, I think, just quite a few years ago. So I'm going to just go through this very quickly. We know that, PARylation is actually very tricky to study for several reasons. One is that it's actually a very unstable modification. What it means is that, when you actually try to sequence that in the mass spec, this adenine moiety, the pyrophosphate bond, and the ester bond, they kind of decompose, you kind of lose the information. The second thing is topologically, PARylation is actually very heterogeneous. The chain length of these modifications can extend to up to hundreds repeating units of ADP ribose. So it's actually a very long chain, a polymeric type of PTM. And it's also very acidic, so it's actually very hard to ionize these modified peptides in mass spec. So again, several years ago, we developed this chemical biology trick where we realized that the first ADP ribose that's being conjugated to proteins is actually via this ester bond for the D and E residues. And we can use a chemical reagent which is called hydroxylamine, attacking this site, and essentially convert a modified D or E residue into a hydroxymic acid. And this chemical group is not found in nature. So all we have to do is just scan the entire proteome and to see which acidic residues, D or E residues, has this extra NH group, and we can tell that residue was previously modified by PARylation. So this technology has been really powerful, and a lot of papers have been published. So we have utilized this technology, identify thousands of sites of PARylation cells. We found that these proteins are involved in many kind of different biological processes, not only DNA repair, but also epigenetic regulation, transcription, and even RNA metabolism. We use this technology in a quantitative fashion, and we studied the PARylation landscape that's actually modulated by clinical relevant PARP inhibitors. And in doing so, we identified many targets of PARP1 and the other enzymes. We have also defined the PARylation landscape for breast cancer, look at how the heterogeneity associated with the PARylation proteome. And then we have also collaborated with folks like Junjie Chen and previously Xiao Chen to look at PARylation function on specific single molecules. So again, all these papers have been published, so I'm not gonna get into details for these studies. But I do want to mention one recent study we actually performed. This was actually something quite interesting with the goal of understanding the potential function for PARylation really on a global scale. So we previously actually looked at some of the PARP modified proteins identified through our studies, and they were actually involving in regulating protein-protein interactions, or PPIs. And we actually was fortunate to actually collaborate with Chen Cong. She is actually really fantastic computational biologist, specialized in using AlphaFold and other AI tools to study PPIs in silico. So 3 years, Chen actually has put together this amazing array of protein complexes models, not single protein complex model, utilizing AlphaFold and other prediction tools. And we collaborate with Chen, asked the simple question is that because we know all these modified sites on the different proteins, if we map all the PARylation sites on these protein complexes, can we garner information about how this modification can potentially regulate PPI networks? And I think the results are actually quite remarkable. Here I'm just giving you 3 examples out of really hundreds of these very exciting models. Here involving a common protein PCNA binding to different binding partners FAN1, XPC, and POD4. So you can see that the partition sites, because we know exactly where partition sites are, we can map them among these protein complexes. And you can see many of these protein complexes actually have the partition sites really on the interface between these protein complex, suggesting that partition actually might play a role in regulating this very dynamic PPI. So right now obviously we're actually taking a biochemical approach to validate some of these results, but extending really onto many interesting protein interaction networks that are regulated by this very important modification. So I'm going to actually quickly jump onto the next concept, which is actually PARP trapping. And as we know that through years we have always been very intrigued by this concept. So this concept was actually first proposed by Yves Pommier, NCI, which he argues that maybe the reason why PARP inhibitors can kill cancer cells is not only because of inhibition of the PARP1 enzymatic activity, but really through this gain of function model of PARP trapping. And at this point, I think a lot of us should be very familiar with this model where when you inhibit PARP1, then you actually cause the PARP1 to be stuck at the DNA lesions. And this can actually be visualized by this laser microirradiation assay. You basically transfect cells by GLP-PARP1, and you can see that when you shine a laser across these cells into this DNA damage, you can see PARP1 is quickly recruited to the DNA damage site. But when you add a PARP inhibitor, in this case rucaparib, you can see that PARP1 becomes stuck at the DNA damage site and then kind of linger around for a longer period of time. So this is actually a classical way of visualizing PARP trapping. Now we know that PARP trapping could have a lot of different function consequences. The replication fork could be collapsed or interfered as a result of trapping. DNA damage could be increased as a result of that. And from a function point of view, trapping actually is known to cause toxicity, as shown by Shanja's recent PNS paper 2023. So this all kind of argues the very important function role of trapping in the context of understanding mechanism of action for PARP inhibitors. So several years ago, we tried to ask a very important question, because again, when we think about these PARP inhibitors, at least the ones that are approved by FDA, they do 2 things, right? They all inhibit PARP1. At the same time, they all trap PARP1, right? So how do we assess the relative contribution of inhibition versus trapping in terms of mediating anti-cancer activity? So this is something we have been always been very interested in. So how do we actually address this question? So several years ago, we actually decided to take a chemical biology approach where we utilize this PROTAC strategy. I mean, I think everybody should be very familiar with that, pioneered by Craig Cruz and others. Is the idea of converting an inhibitor into a degrader by this PROTAC strategy. And essentially, what PROTAC means is that you can basically attach a E3 ligase recruiter. In most cases, you can use a ligand that binds to cereblon, or you have another ligand binds to VHL. These 2 are very commonly used E3 ligases. But when you actually link the inhibitor with this E3 recruiters, you essentially create these heterobifunction molecules that can actually help to recruit E3 ligase to degrade a protein of interest. So we carry out this PROTAC campaign, and obviously there's a lot of work context of medicinal chemistry, design, and everything. But finally, we were able to actually get this molecule. So again, for any of you who might be interested in learning more about PROTAC, I'll be happy to talk about it after the seminar or offline. So this is actually the structure we were able to actually derive. The compound actually has 3 components. This is actually the from the rucaparib, which is again FDA approved PARP inhibitor. This is actually a portion of pomalidomide, which actually is responsible for recruiting an E3 ligase called cereblon, and you basically link them together by an OPAC6 linker. This was actually done in collaboration with chemist Chuo Chen's lab back at Southwestern Medical Center. So this compound was basically the best compound we were able to derive, and the compound was actually extremely potent. if you look at the degradation profile of this compound in primary cardiomyocytes, you can see that even at 50 nanomolar, you can see a robust degradation of PARP1. Considering PARP1 being extremely abundant nuclear enzyme, we thought this result was pretty remarkable. And if we compare the PROTAC compound irucaparib compared to the parental compound rucaparib, you can see in both cases you can see strong inhibition of PARylation by both compounds. But the key difference here is that the regular PARP inhibitor inhibit PARP1 without changing the PARP1 protein abundances, but the PROTAC compound is able to inhibit PARP1 at the same time causing PARP1 degradation. And we also show that this compound is extremely selective. Within the entire proteome, this compound really only degrades two proteins, PARP1 and PARP3. Now, the reason why this is actually important is now we can actually use that as a tool compound to understand, really several key features, comparing the inhibition of and also trapping, right? So if you look at the PARP1 laser microirradiation assay here, you can see that if you treat cells with a rucaparib, you can see that potent PARP1 trapping. But if you treat the cells with irucaparib, the PROTAC compound, you can see there is a greatly diminished total PARP1 pool, and as a result of that, the trapping of PARP1 is actually greatly reduced. So now we actually have these two compounds. One is actually a degrader, which is inhibit PARP1 without trapping. And then you have another compound, which is the inhibitor, which, the regular inhibitor, which causes both inhibition as well as trapping. So if we compare the cell biologic function for these two compounds, you can see there is actually tremendous difference. So the regular inhibitor, you can see there is a, cell cycle disruption and also cell death, which is not found when we treat the cells with the PROTAC compound. And we have published a series of papers. Again, I'm not going to get into details, but the conclusion is that the PROTAC compound does not induce, obviously, trapping in DDR, does not induce apoptosis. We have also recently shown that regular PARP inhibitors cause activation of the cGAS-STING pathway. However, the degrader actually does not induce the activation of the innate immune signaling. And finally, we recently showed that the degrader also does not cause the inflammatory cell death, which is apoptosis. So all these results are actually consistent with the model, at least for the system we have tested, that trapping is actually a key driver for the anti-cancer activity of PARP inhibitors. And also we have done some further work just to understand the immune modulatory roles for these compounds. Excuse me. As you can see that these compounds are actually if we compare 5 different PARP inhibitors, and you can see they have similar inhibitory activity for PARP1, but they cause very different degree of PARP1 trapping, TALA being the most potent PARP1 trapper, and veliparib is actually the weakest PARP1 trapper. And you can see the degree of the endemic response as well as the degree of the cGAS-STING pathway activation, all kind of correlated with the degree of trapping, right? And obviously we know that these 4 PARP inhibitors, TALA, niraparib, rucaparib, olaparib, they all have been approved by FDA, but veliparib is actually still being studied in a clinical setting. So I would say that these results are consistent with a model that maybe trapping is something we want to kind of pursue to increase in the context of anti-cancer activity. Now, one thing we have been very curious about is that what exactly is trapping? What is the molecular underpinnings for PARP1 trapping? And this one is actually a recent story we kind of finished. So I'm gonna share that story with you a bit more in detail. So to understand the molecular underpinnings of PARP1 trapping, we actually did a screen. So the screen was actually based on understanding how the proteome is regulated at the DNA damage site. Okay, so here we design experiments where we basically treat cells with all these different conditions, hydrogen peroxide or MMS, which are known to activate PARP1 very potently. Then we treat the cells either in the absence or in the presence of talazoparib, which is a very potent PARP inhibitor. And then what we do is we basically biochemically isolate the chromatin fractions from these cells, and then we actually label them by the TMT reagent, which is actually isobaric labeling strategy, to perform quantitative proteomic analysis, which is something we have done quite a bit, in the previous work. So the idea is that we want to measure protein abundances in all these different nuclei, the chromatin fractions, and to understand, what are their protein dynamics, either regulated by PARylation, on and off, so this kind of dynamics we're really interested in understanding. So obviously, this is actually a very large study. So we're able to identify many different proteins. So here I think the results actually argue that these proteins can be classified into different groups, four different groups. So I would like to actually draw your attention to group 1, which actually represent the proteins have this following dynamics. So in control cells, the protein is actually more or less flowing around in the nucleus. But when you actually trigger DNA damage by hydrogen peroxide or MMS, again, remember these two are very potent activators of PARylation. Then you see the protein actually being recruited to the damage site. Now, in contrast, if you pre-treat the cells with a PARP inhibitor, then the recruitment of this protein A is abolished, right? So that's kind of consistent with the model where this protein A presumably binds to poly-db-ribose and gets recruited to the DNA damage site in a PARylation dependent manner. So if you look at some of the proteins actually we found in this group, you can see proteins like XRCC1, LIG3 and others. These are actually known proteins that are bound to poly-db-ribose. Again, arguing the validity of this assay. So when we're actually going through this list of proteins, we got intrigued by this very interesting protein called RNF14. Okay, and you can see this protein again was enriched in the hydrogen peroxide MMS treatment group, but then the recruitment was actually abolished if you pre-treat the cells with talazoparib, right? So this is actually again is consistent with a PAR dependent protein factor. So and the other reason why we chose this protein to study more mechanistically is that besides this protein was actually a very poorly studied protein, is that those protein actually contains very interesting function domains. So if you look at this domain structure for RNF14, you can see a classical ring domain which is consistent with it being a ubiquitin E3 ligase. It also has this so-called tandem C2H2 domain, right? And such domains actually have been found previously in several proteins that are known to bind to poly-db-ribose. And in fact, people actually sometimes call these tandem C2H domain to be called a PBZ domain, which stands for PAR binding zinc finger motifs. Okay, so at that point, we kind of immediately tested the hypothesis that maybe this protein could be a PAR binding protein. And when we actually performed this kind of dot blot assay, we found that the wild type protein RNF14 indeed actually binds to poly-db-ribose pretty well. And if you actually mutate several key cysteine residues in either the first or the second or both C2H2 motifs, you abolish the PAR binding. Okay, so that argue that these two domains are very important in terms of PAR binding activity. At the same time, we can also do this cell biology experiments where we basically knock out RNF14 and then re-express either the wild type protein or the protein with mutation in the PBZ domain, the key cysteine residue. You can see again that this protein was enriched in the chromatin bond fraction, but was abolished as a result of tala treatment. The mutant protein actually was not enriched. So all these kind of results are consistent with the model that this protein potentially is a PAR binding protein. Now, what does it do? I mean, when it's recruited to the DNA damage site, what exactly is the function? Again, as I mentioned earlier, this is actually really a poorly understood protein. And we did actually IPMS experiments and just to see what is the binding partner for this protein. And, very interestingly, we found that PARP1 actually binds to RNF14 actually very, very tightly. And it seems that the interaction between PARP1 and RNF14 is mediated by poly-db-ribose, because if you pre-treat the cells with a PARP inhibitor, then the binding was abolished. So that's again is consistent with the model that PARP1 is probably like one of the most heavily parted protein in cells. So PARP1, when it's activated, it just modifies itself like crazy, right? So this is actually consistent with our model. And then what we also did is that we found that RNF14 really does a very good job in terms of mediating ubiquitination. So if you actually pull down PARP1, you can see that the ubiquitination level for PARP1 is actually greatly reduced in cells with RNF14 knockout. And you can see the total PARP1 with the PAR form is actually looking at only the PARP1 form is also actually reduced as a result of expressing RNF14. If you knock it out, then this form is actually accumulating. So at that point, we come up with a hypothesis that maybe RNF14 targets PARP1 for ubiquination and degradation. And again, remember, PARP1 is actually really is enriched with at the DNA damage site. So we feel like maybe RNF14 plays a very important role in regulating dynamics of PARP1 at the DNA damage site. Now, why that's important? Because again, as I mentioned earlier, this is actually highly relevant to the trapping phenomenon we just kind of described. So here actually we did some more genetic experiments. Here what we did is we actually generate again the RNF14 knockout cells and reconstitute these cells by either the wild type RNF14 or the RNF14 mutation in the PBZ domain or the RNF14 mutant in the ring domain. Again, keep in mind that the ring mutation again is assisting in the ring domain is actually very important for its E3 ligase activity. So if you mutate that site, the ring domain is inactivated. On the other side, if you actually mutate the key cysteine residues in the PBZ domain, you actually lose the PAR binding activities, right? So then we can actually look at the PAR dynamics in all these different three different cell contexts. As you can see that in the cells expressing the wild type RNF14, you kind of see like this kind of normal dynamics of PARP1, which gets recruited very quickly, 5 to 10 seconds. Then by 2 or 3 minutes, you kind of see gradually decreasing level of PARP1 at the DNA damage site. For cells expressing the RNF14 mutant in the PBZ domain, you can see PARP1 now actually start to linger around a bit more, right, compared to the wild type. And again, that's arguing that if you're unable to recruit RNF14, because again, the mutation in the PBZ domain, then you actually disrupt the PARP1 removal. So that's actually consistent with the model. But I think the most striking kind of phenotype come from the cells expressing the ring domain mutant. In this case, you can see a very dramatic PARP1 trapping phenotype. And I think this is actually consistent with the dominant negative effect, which is that you're still able to recruit RNF14 if you have a ring mutation because the PBZ potentially is intact. But when you recruit a RNF14 mutant, that's a dead enzyme because it's not able to remove PARP1. So that's causing a very profound trapping phenotype. Right. So this is actually all the genetic evidence. So let's actually kind of summarize everything we talked about so far. That we identified this novel protein E3 ligase as a PAR-dependent DNA repair factor. It targets part of PARP1 for ubiquination and potential degradation. And RNF14 ablation or ring domain mutation leads to protein PARP1 trapping, right? So that's all the kind of mechanistic information we have garnered from the study. I think a very unique feature about this pathway is its potential drugability, because again, remember all the inhibitors we talk about have this very important trapping activity. So one thing I always kind of mention to the trainees in my lab is that we don't want to stop a study at the publication. We always want to think about what is the therapeutic potential, right? So I think a very unique feature about this pathway is that it's actually druggable. Sorry about the voice in the background here. So actually several years ago, we came across with this very interesting paper from Dan Nomura's lab, published in Nature Chemical Biology, that they found this natural product. It's called nimbolide. So nimbolide is actually found in the it's called a neem tree, which is found in the Southeast Asian countries, India, Bangladesh, Malaysia, and others. So the leaves and barks from this tree actually contains a very high concentration of this compound called nimbolide. And for those of you who actually who might be chemists, can tell that this compound is actually a mycoreceptor, right? So it has this mycoreceptor right here. And indeed, Nomura's lab showed that by reacting with a very important cysteine residue of RNF14, this compound potentially could be RNF14 inhibitor. Of course, back then the function of this protein was actually not known. So we test this strategy. Actually, it turns to be the compound is actually very interesting. So if we perform in vitro ubiquination assay, you can see that RNF14 is able to modify itself by ubiquination, as shown by this ladder here. Now, if you actually add nimbolide in this in vitro ubiquination assay, you can see that the ubiquination is actually potently inhibited by the addition of nimbolide, right? Now, if you now actually look at the cell biology assay I just mentioned, the GFP-PARP1 laser microirradiation assay, you can see that nimbolide consistent being an inhibitor for the E3 ligase activity for RNF14 cause very, very potent PARP1 trapping, right? So you can see the level of PARP1 trapping is similar or even greater compared to olaparib, which is a well-established PARP1 trapper. So at this point, we actually have two strategies. One is actually genetic method, and the other is actually a pharmacological method to perturb this pathway and to actually cause very important PARP1 trapping. And as we kind of move along, we kind of think about something else, which is actually we want to think whether it's actually possible that the trapping of PARP1 as caused by regular PARP inhibitors versus nimbolide could be different. And the motivation for this is, again, as I mentioned earlier, the reason why PARP inhibitors called PARP1 trapping is you're blocking the enzymatic activity of PARP1. So you're actually potentially eliminating the degradation mechanism for PARP1 as mediated by RNF14 and other mechanisms. So that's actually one mechanism for causing PARP1 trapping. Now, a unique mechanism of causing PARP1 trapping is not interfering with PARP1 itself, but rather interfere with a upstream regulator, in this case, RNF14, because this is actually precisely what we do, is to inhibit the enzymatic activity of RNF14 by nimbolide, right? So we want to see whether you have a different kind of trapping phenotype compared to these two different mechanisms. And we actually look at a classical poly-DP ribose binding protein, which is XRCC1. And as you can tell, that nimbolide treatment actually causes a very potent XRCC1 trapping. In contrast, you can see that regular PARP inhibitors or olaparib, by inhibiting the enzymatic active PARP1, you can see that the trapping is not found in this case. So at this point, we can argue that maybe these two mechanisms cause very different trapping phenotype, and we can actually recapitulate that by a different assay, independent chromatin fractionation assay. As you can tell, that nimbolide causes very potent PARP1 trapping, and it also causes unique XRCC1 trapping, right? So this is actually very different from regular PARP inhibitors. So here, just to kind of summarize everything, we have identified a novel and very unique PARP1 trapping pathway that's regulated by this E3 ligase called RNF14. And the trapping caused by nimbolide and the other potentially RNF14 inhibitors is actually different from a regular PARP inhibitor because this is a part of PARP1 and potentially PARP binding DNA repair factors like XRCC1. So this is really arguing that the trapping caused the accumulation of PARP1 and then the other protein factors as a super trapping complex. And again, the nimbolide is actually a gain of function. So when you have this RNF14 being covalently modified by nimbolide, you actually introduce this new activity, which is not found in nature. And we have also done work, where we found that nimbolide is able to synergize with other DNA, DDR compounds. Actually, nimbolide is able to introduce, very good, potency to kill BRCA deficient cancers. And I think, this is something I want to highlight here, which is, obviously we know that from a clinical point of view that, many patients actually don't respond to PARP inhibitors. And for those who actually do respond, a large fraction of them actually eventually develop PARP inhibitor resistance. So PARP inhibitor resistance is actually a really major problem in the clinic. So here we actually test a couple models using our nimbolide compound. You can see that HCC 1937 is actually a classical BRCA1 deficient breast cancer cell line. This cell line is known to be resistant to regular PARP inhibitors. And as you can tell that this cell line nevertheless is still very sensitive to nimbolide, both in vitro as well as in animals. And we also test nimbolide in different contexts of cryo resistance by testing this subclone, which was obtained from Lizo's lab. So at this point, we actually have this very interesting mechanistic insights, and then we start to collaborate with a very good colleague, Tian Qing, who is really a fantastic synthetic chemist. And as you can tell, this compound nimbolide is extremely complex. It has 5 different rings and a whole bunch of stereocenters. It's a very, very complex natural product. But Tian was able to derive a total synthesis route for this compound. By doing this retrosynthesis analysis, you can basically cut this molecule into 2 halves. This microacceptor region is very important for its activity, but you can actually change on this portion. So together with Tian, we actually have, synthesized and also evaluated more than 300 analogs of, nimbolide, where you can see, like, by changing different, part of the molecule, you can actually increase the activity. You can actually derive, analogs with improved potency and drug-like properties, and we can actually do target revalidation, using these novel analogs. So at that point, Tian and I, we actually co-founded the company, really trying to push, nimbolide analogs for preclinical studies and hopefully clinical studies. So we're actually very excited about this program. So I think, the last 10 minutes-ish, I want to kind of just talk about yet another new study that was just published a couple of months ago in the lab, where we're really interested in understanding the potential utility of PARP inhibitors, hopefully beyond BRCA deficient cancers. And as we know that BRCA deficient cancer, has been, the treatment for these cancers have been really kind of dramatically revolutionized by PARP inhibitors and others. But at the same time, there's actually a strong appreciation that maybe some other tumor types might also respond to these compounds and benefit from these agents, right? So here I want to introduce a very interesting cancer type, which is called small cell lung cancer. So you probably heard of this non-small cell lung cancer, which is really the major subtype of lung cancer. But again, about 10 to 15% of all the lung cancer patients are actually coming from small cell lung cancer. Now, small cell lung cancer is actually completely different from a biology point of view, and also histology and others from non-small cell, because they're just very different cancers. Okay, small cell is actually a highly malignant human cancer. The 5-year survival is about 5%. It's one of the most malignant human malignancies out there. If you look at the mutation spectrum for small cell, it's completely different from non-small cell. All the small cell lung cancer, have the P53, or a very, very large fraction have P53 and RB mutations. Small cell typically does not have the mutations found in the small cell, like KRAS, EGFR. This is actually very rare. Also, BRCA mutations also now commonly found in small cell lung cancer either. Now, small cell usually are pretty sensitive to chemo and also radiation, but almost invariably the patients will develop resistance, and then at that point there are actually very few therapeutic options. So I think for small cell lung cancer there is critical need for additional therapeutic options for these patients. Now, several years ago, a colleague back at Southwestern, Ben Trapkin, actually reported the clinical response of small cell lung cancer to actual PARP inhibitor. This was actually something very, very exciting. So suggesting PARP inhibitors could be useful for this cancer type. And we actually started to collaborate with Ben Trapkin and also John Minna, because all the reagents they have are actually very unique. So we actually put together a panel of small cell lung cancer cell lines, 24 cell lines. Now here, this is something very important to remember, that small cell lung cancer cells, just like non-small cell, is highly heterogeneous. Okay, and they can be classified into different subtypes based on the expression of certain lineage-specific transcription factors. So ASCL1 is actually a major lineage-specific oncoprotein. NeuroD1 is another one, and POTF3. So basically, you can classify the small cell tumors based on expression of these lineage-specific oncoproteins. Okay, and for example, ASCL1 is actually a major one. It's actually a very important survival factor. It's also maintaining the neuroendocrine differentiation for small cell, which is actually was reported by Minna and the colleagues several years ago. And at that point we actually become very interested. So we put together this cell line panel, about 24 cell lines that represent the major small cell lung cancer subtypes. The ASCL1 subtypes are highlighted by this kind of brownish color. The bluish color is actually NeuroD high, and you have the POTF3 and also the others, right? So these are actually a representative of the typical small cell lung cancer landscape. And then what we did is we asked the question, okay, so if we actually treat these different small cell lung cancer cell lines by a PARP inhibitor, in this case talazoparib, and then we asked the question is that how does that change the global proteome or the protein expression for these cells? And we want to understand the mechanism of action for PARP inhibitors in these, and trying to understand which might respond and which might not respond to PARP inhibitors, right? So this is actually, as you can tell, it's a very large experiment involving 24 cell lines with or without the treatment of a PARP inhibitor. we perform global protein expression analysis using the TMT platform, and we were able to quantify more than 12,000 proteins in total. So it's a very, very large scale experiment. And again, we're kind of really focused on events that are regulated at the post-transcriptional level, because again, we're measuring protein abundances. So we pay attention to a lot of the things actually happening after the transcription and post-transcription. So again, this is actually a hugely useful resource, and we start to perform lots of bioinformatic analysis. And here, I just kind of want to just distill the information down to a couple key messages here. One is that we found that if you compare the protein expression changes in ASCL1 cells, for example, ASCL1 high cells, okay, and you can classify them into sensitive versus resistant. So all of them actually express ASCL1, but some of them actually are sensitive to PARP inhibitors, some actually are not. Now, if you look at the total protein expression changes in this group, it's actually very, very different from this group, okay, meaning that there are many proteins whose expression is now remodeled or changed as a result of PARP inhibitor treatment, only in the sensitive cell line, but not in the resistant cell line, right? And then we actually look at all these things and perform gene ontology analysis. A lot of them actually are very important for cell cycle regulation, cell growth, which is consistent with the phenotype we look at. But among all the proteins that show a downregulation as a result of PARP inhibitor treatment, we actually pay attention to a couple, which is actually this very interesting ASCL1 protein I just mentioned, which is a key lineage-specific oncoprotein I just mentioned, right? So then we actually perform all the function studies to validate our finding. And obviously, first is that if you take a small cell lung cancer cell line, if you treat the cells with talazoparib, you can see a very robust ASCL degradation, both in vitro as well as in xenografts, right? So in both cases, you can see this consistent results. And this regulation is actually happening on the post-transcription level, because if you look at the MR level for this gene, it actually doesn't change before and after PARP inhibitor treatment. Now, as I mentioned earlier, one of the very unique features is that small cell usually is very sensitive to chemo, as Lizo at the first line of therapy. Now, if you look at the ASCL degradation phenotype, it's actually recapitulated by the clinical relevant chemotherapeutic agents, etoposide, doxorubicin. So these are actually commonly used to treat small cell lung cancer patients. And you can see that ASCL1 is also degraded as a result of the treatment of these chemos, right? And it's actually surprising that after so many years of these chemos being applied, we actually don't know exactly why small cell responds so well to chemo, at least the first round. But I think ASCL degradation actually proposed a very interesting mechanistic explanation for this finding. And also consistent with these results, we found that you can see there's increased level of ubiquination for ASCL1 that's isolated from cells treated with TALA. So again, that's arguing that the degradation of ASCL1 probably is happening at the UB level. Now, the question here is, what exactly is E3 ligase? Now, here I'm going to refer to this very interesting E3 ligase called HUE1. And the reason why we're interested in this HUE1 E3 ligase is for several reasons. One is that this E3 ligase is known to respond to DNA damage signal. Okay, so this is actually a DNA damage responsive E3 ligase. Then regular ubiquination of many downstream targets involving cell death and DNA damage response and others. So this has been well documented in the literature. There's another very interesting paper in Science, 2016, where they actually look at the connection between HUE1 and ASCL1 in context of neuronal stem cells, so not in small cell lung cancer, but in a different context, neuronal stem cell. And they found that HUE1 is actually responsible for degrading ASCL1 in that context. So we immediately come up with a hypothesis that maybe HUE1 can play a role in this case to mediate the PARP inhibitor-induced ASCL1 degradation in small cell lung cancer, right? Now, the first thing we want to test is that, we found that, by looking at the ubiquination, or ubiquination of HUE1, you can see a dramatic increase in cells treated with TALA. So this is consistent with the HUE1 being a DNA damage responsive E3 ligase. And also, if we look at some of the known HUE1 targets like MCL1, which is a critical apoptosis regulator, you can see a very robust degradation of this target by HUE1. So at this point, we can say pretty confident that HUE1 is definitely activated as a result of TALA treatment. And the question is that how does ASCL1 fit into that kind of paradigm? So if you knock down HUE1 in this case, then you can look at the total UB level for ASCL1. You definitely see a very robust decrease, right? So in this case, we can argue that HUE1, very likely plays a role in regulating ubiquination of ASCL1. If we knock down HUE1, then you can also see that the degradation of ASCL1 now is actually very much blocked, even treating cells with talazoparib, right? So this is actually consistent with this E3 ligase play a role in degrading ACL1. And you can see that by reducing the HUVO1 level, you're also reducing the sensitivity of these small cells to talazoparib. So you can see there's less cell death as a result of HUVO1 knockdown in the response to talazoparib treatment, right? So this is actually based on multiple experiments. So here, we would like to argue that this E3 ligase is very important in regulating ubiquination and also degradation of ACL1. And again, because the ACL1 is so important in regulating survival and proliferation others for small cell, regulation, depletion of HUVO1 also can protect cell death against PARP inhibitor treatment. So moving on to some of the biomarker studies. So one thing I want to kind of argue here is that, as you can see, that if we think about the entire flow chart here, we treat cells with PARP inhibitor. PARP inhibitors will always cause trapping. No matter what cell lines you test is, no matter what the context is, you'll then induce DNA damage. You probably will induce HUVO1 activation. So everything up to this point will be the same, regardless of the cellular context. Now, as I talked about earlier, if you look at the different cell lines or clinical response, not all the small cell lung cancer patients respond to PARP inhibitors, right? So there are patients or cell lines that respond, but some of actually don't respond. So the question here is that why the signal is the same up to this point, but then you see different response to this signal. This is actually obviously a very important question, not only at the basic biology side, but also moving to the clinic. Can we actually come up with predictive biomarkers for PARP inhibitors for small cell lung cancer patients, right? So this is something we actually spend a lot of time, think about and also testing. So I want to kind of draw your attention to this data here. So, we put together a panel of small cell lines here, not as extensive previously, but, nevertheless actually is representative. So you can see all these cell lines. There are actually several that are, very sensitive to PARP inhibitors. There are actually a couple that are, really resistant to PARP inhibitors. Now, as I mentioned earlier, if you treat the cells by PARP inhibitors, you will see gamma H2X, which is a marker for DNA damage in all the cell lines tested, right? So this is actually, again, consistent with our model here is that the DNA damage signal is universal. It's a general mechanism. But I think the difference is that how the different cells respond to that. And here, I think the data is actually extremely interesting is that if you look at all these cell lines that are sensitive to resistance, you can see that in sensitive cell lines, you can see very robust ACL degradation in all these 5 different cell lines. But in the resistant cell lines, you can see the ACL1 is not degraded in response to PARP inhibitor treatment. And also consistent with that, these cell lines are actually very resistant to PARP inhibitor treatment. Now, this data actually is extremely interesting because it argues that PARP inhibitor generates a common DDR signal, but different cells, actually different small cell lung cancer cells, respond to that signal differentially, right? So that's actually, I think, where the context becomes very, very important. Now, what's the difference between these two set of cell lines? I think, one of the things we immediately ask is that how does the HUVO1 expression look like, among these two different cell line groups? As you can tell, the sensitive cell lines express a good amount of HUVO1, but the two cell lines that are resistant are absent for HUVO1 expression. And also, more interestingly, we can actually force HUVO1 expression in these two resistant cell lines. So in this case, we transfect the cells, by virus, HUVO1, and you can see the HUVO1 now is starting to express in these two cell lines. Now, if you treat the cells with talazoparib, you can see actually an appreciable level of ACL1 degradation, and the cells actually start to die. I mean, it's not complete death, but you can see a good amount of cell death in this case. So essentially, by expressing HUVO1 in these resistant cell lines, we can convert them into sensitive cell lines, right? So that argues that maybe HUVO1 expression could be a predictive biomarker. So we actually dig into the previous clinical study. This is actually a paper from George et al. In Nature 2015, where we can segregate these patients in their response to chemo. There are no clinical data for PARP inhibitor, unfortunately, but there are data for chemo. So we can actually segregate them into 2 groups, HUVO1 high and also HUVO1 low. And as predicted, you can see that the patients actually respond better to chemo if their tumors express high level of HUVO1, again, presumably by degrading ACL1, right? So that's actually that. So here, I think, if we think about all these data together, we can, argue there could be a potentially novel synthetic therapy model. We all know this very well, that BRCA mutation predicts, response to PARP inhibitor. This is a very well-established synthetic lethality model, but I would argue that maybe this could be another synthetic lethality model based on the expression of ASCL1. So again, the PARP inhibitor DDR signal, when it happens on other cell type, probably the cells, don't care or don't care too much. But if you actually have small cells, especially those ones that express ASCL1 and dependent on ASCL1 for survival, and plus, expressing inhibin-1, then this signal is going to be converted into a cell death signal and causing the death of these small cell lung cancer cells, right? So essentially, this could be a novel mechanism to create a synthetic lethality and hopefully a therapeutic window for these patients. So we are actually working with Ben Drapkin now to actually really kind of testing that hypothesis using clinical relevant samples at this point. So just a kind of quick summary for the PARP inhibitor small cell, we have performed global proteome profiling. We look at a mechanism of action for ASCL degradation, and then we come up with a predictive biomarker, and hopefully we can actually have more kind of clinical relevant data in the near future. So kind of summarize for everything I just talked about today. Again, we have been very much focused on the mechanism of action studies for PARP inhibitors, and this really kind of come from 3-4 different angles. One is looking at the inhibition of PARP1 and other PARP enzymes. This has been really facilitated by the proteomics technologies we previously developed. We look at PARP1 trapping. I didn't get a chance to talk about too much, but we can reduce trapping by PROTAC compound. We can increase the trapping by this novel pathway, a unique pathway of RNF14 plus the therapeutic strategy of nimbolide. And then we're really excited about the potential expansion of the PARP inhibitors beyond the BRCA deficient cancers, hopefully to small cell lung cancer and other solid tumors. So I want to really acknowledge the people who did this work, Yajie, Yuan Li. They're actually involved in the initial development of the PARylation proteomics work. Shuai actually previously worked on the PROTAC technology and trapping. And Peng and Chiho has been really kind of heavily focused on the recent work in the nimbolide project and also the small cell lung cancer project. So again, really want to thank their work. We're actually really fortunate to collaborate with many fantastic collaborators. Tian, as I mentioned, a synthetic chemist, Chong Chen, Jiang Mena, Ben Drapkin, Ling, I didn't talk about too much, but she's a computational biologist. Corey here, and also Shan for the wonderful collaboration as well. So finally, want to argue that really there's so much to be learned in protein modifications, and hopefully I can convince you that today what we're talking about is just the tip of the iceberg. And a final word, I just want to put a plug. We're actually recruiting folks. So if you're interested in DNA damage, cancer biology, and drug discovery and chemical biology, please feel free to contact me. Thanks again. I'll be happy to take questions.
That's great. So yeah, if anyone have questions, please raise your hands, and I would love to active them so that you can ask him directly. So I just have a quick question. This HEB1 is X chromosome genes that in males there's only one copy of them. Do you see any association with gender difference in terms of the sensitivity?
Oh, you know what? That's actually a very important point. We actually we didn't realize that the gender difference for this gene. So obviously, when we look at the expression level for these different cell lines, we saw cell lines expressing high level of HEB1. Obviously, there are cell lines expressing low levels, but we did not look at their history. But I think this history actually is known. We can go back and look at ATCC data. But thanks for that. I think this is something very important to consider.
Yeah. So Gerald.
Yes. Hello.
Yeah.
Nice talk. Very good. I have a question about WEE1 and its recruitment to the target protein. I saw it as a WWE domain, so it should bind poly-ADP ribose. How do you explain the recruitment if you inhibit the poly-ADP ribosylation of PARP1 by the talazoparib? Do you have a mechanism of action?
Yeah, this is actually something very interesting. So previously, this project we have been working on for actually quite a few years. So I think in the initial stage, we're kind of debating about two different pathways moving forward, right? So one is that, as you mentioned, that HEB1 could potentially be recruited to target protein via binding of its WWE domain to its polydp ribose. We're actually testing that hypothesis using some of the model proteins. ASCL1 is a bit harder. I mean, because the PARylation on that protein, we have a very hard time detecting that. So because of its amino acid distribution, we can talk about this on the biochemical side later. So, we kind of testing this hypothesis actually for quite a bit, and we cannot find evidence that there is potentially binding between HUE1 and ASCL1 in a PARylation dependent manner. Okay, so this is something we don't have evidence for. Now, is it possible that the recruitment of HUE1 to ASCL1 is through some other mechanism? We feel like that's probably more likely. Now, the issue here is that we don't know exactly what that mechanism is. It could be, through phosphorylation, through some other DNA damage signal, PTMs. I mean, it's just a lot of possibilities, and quite frankly, we don't know at this point what exactly is that critical signal that brings these two proteins together. So we don't know at this point.
Okay,
Yeah,
thank
so
you.
we know quite a few questions. So Chris, you want to ask it live?
Thank you. I hope you can hear me. So it's very nice. I have two questions. So the first is that BRCA1's a known substrate of HUE1, and a number of people have shown this. So have you excluded the possibility that the effects you see with HUE1 are not in part at least controlled by controlling levels of the BRCA1 protein?
That's again another very important point. We have not tested that hypothesis, but we can actually obviously go back and look at that possibility. Yes.
Okay, thank you. And my second quick question is, it's interesting that nimbolide works in HCC 1937s, which I think, one hypothesis is that resistance is because they also lost one of the shielding proteins.
Yeah.
But does nimbolide work in models where you have a BRCA1 or BRCA2 reversion, which looks like to be the more dominant mechanism of PARP inhibitor resistance?
So, Chris, this is actually another very important point. We're thinking about testing this, compound in different resistance contexts. So far, as I mentioned in the presentation, we have tested, in two models. One is that the HCC 1937 we just talked about, which is a de novo resistant mechanism. The other one is actually this SYR12 UWB1 subclone from Li Zhou's lab, which is actually a cryo-resistant model. In both cases, we can see that nimbolide is able to kill these resistant cells. We have thought about using more models in context of BRCA mutation reversal, but unfortunately, we don't have access to these cell lines. So if you know where to get them, I'll be more than happy to actually test them. If you can share the information.
Yep, we have them. We published them. You're very welcome to use them.
That'll be awesome. Thank you.
Thank you.
Paula, you want to ask live? I think she disappeared. Paula? No? Okay. Jean-Philippe?
Yep.
Yeah, go ahead, please.
So do you have an idea what is specific to the C2H2 zinc fingers of RNF114 regarding PAR binding that would not be present in the hundreds of other C2H2 type zinc finger proteins?
Yeah, you know what? That's actually another very important point. So, I mean, we haven't got a chance to study the biochemical side for this too much at this point. But I do feel like, just because these C2H domains are tandem, right? So if you think about these are two domains that are right next to each other. So, it is possible that, these two domains are actually responsible for binding to PAR chains with longer chain length. This is actually a hypothesis. We haven't got a chance to actually test that. If you look at some of the early papers looking at, similar PBZ domains in other proteins, they show a very strong tendency to bind to longer PAR chains, but very weakly to mono-ADP ribose or some of the shorter chains. So we feel like there's actually another similar scenario here for this PBZ domain as well. Although again, we haven't tested this extensively using biochemical methods.
Okay, thanks.
Thanks.
Yeah, so there is a question from Hamid Emma asking that, thank you so much for the amazing talks. What is the dose of PARP inhibitor used in the mass spectrometry large-scale experiment, and for how long?
So I assume you're asking these TMT experiments for these studies. We're actually using 1 micromolar. And I think, I believe it was 48 hours treatment, but I can go back and double check with my postdoc.
Yeah, and the Paula is asking that whether nimbolide only covalent attached to RNF114 or possible other proteins in DNA damage response as well.
Yeah, that's another very important question. So again, just because the limitation on time didn't get a chance to talk about this, but we actually have done several important control experiments. If you knock out RNF14, then the cytotoxic effects of nimbolide is greatly diminished. I'm not saying like it's completely gone, but it's greatly diminished if you knock out RNF14. So this data basically argues that this trapping as a result of nimbolide treatment is actually working primarily through the RNF14 dependent mechanism. Again, just keep in mind that RNF14, when you have nimbolide coupled to it as a covalent mechanism, you create a gain of function protein small molecule compound, and that's actually where it conveys really the trapping activity.
Sounds good. I mean, maybe just one more comments before I let you go. If I get this right, I think ASCL1 also highly expressed in neuroblastomas. That might be another area which you can try.
Yeah, absolutely. So this is something we actually have been thinking about as well. ASCL1 is actually neuronal transcription factor, right? So it's involving neuroendocrine cancers like small cell lung cancer, but definitely the other tumor types potentially expressing ASCL1 could also benefit from PARP inhibitor as well. So very important point. Yes.
Cool. Okay. If there's no other questions, thank you very much.
Thank you.
See you guys next week.
See you. Bye-bye.
Bye.